How could it be? For as long as human thought has dwelled on the concept of our own existence, we have been compelled to answer the question of how and why life exists at all. Religions and spirituality have nearly consistently pointed to an invisible, all-knowing, all-seeing creator. Barring the existence of such a being, how could it be that the incredibly dilute and simplest of organic chemical compounds that do exist naturally without organic life coalesced in just the right ways, under just the right conditions, to form the immense order and precision present in even the simplest life forms? Dr. Pepper.
How life on Earth began. Carbon monoxide, carbon dioxide, hydrogen cyanide, ammonia, water, formaldehyde, acetylene, methane, formic acid, methanol, acetone, acetic acid, acetamide, formamide, cyanamide, ethylene glycol, and glycine are among the most biologically relevant of the hundreds of organic chemical compounds that have been confirmed to exist out in the universe where there is no life via radioastronomy. They have been found in molecular clouds trailing comets and asteroids, and could have been deposited on Earth through interplanetary dust particles and orbital bombardments. Terrestrial synthesis by photocatalytic, geothermal, and mineral-driven oxidation-reduction reactions during the millions of years of Earth's prebiotic existence are also an equally likely source. It is unknown from what proportion of terrestrial or extraterrestrial sources the organic compounds came from that brought about life, but it is certain they existed, but most likely in extremely low proportions compared to inert inorganic compounds. The entirety of the biomass on Earth represents 6 times 10 to the 15th kilograms of organic carbon, which, if spread evenly across the Earth's surface, would account for a measly 10 mm thick layer. If broken up into inorganic monomers such as glucose and amino acids and dissolved in the ocean, it would be a mere 5 micromolar solution with 1 monomer for every 10 million water molecules. Living cells have a metabolite concentration in the millimolar range, representing a concentration of over 1,000 times greater. This is known as the concentration problem of life. The second law of thermodynamics teaches us that entropy, or the measure of the possible orientations within a local system in the universe as well as the entire universe, always tends to increase over time. This means that greater concentrations of chemical species within a localized area tend to decrease over time instead of concentrating, and typically require organic life performing the work or some naturally occurring phenomenon to achieve relatively high concentrations. Entropy is both a measure of the possible orientations within a system and the unavailability of energy to perform work. Many scientists believe that entropy will continue to increase in the universe until no meaningful physical interactions can take place in the universe, as all energy available to do work is gone, and the temperature everywhere will be equal. This is known as the heat death of the universe. Now, if you understand the concentration problem of life and the concept of entropy, you might seriously question how life existing at all seems to violate the second law of thermodynamics, as life is inherently a very ordered system, and life creating more life could be seen as fighting against entropy. There are already some good videos out there explaining this, like the one Veritasium has, and I'll give a brief explanation though, and it's that life dramatically increases entropy just by existing. A quick example is that the sun produces immense energy in the form of high energy, short wavelength, high frequency photons, which once they hit the Earth, they'll hit a plant and be turned into chemical energy, and multiple lower energy, long wavelength, low frequency photons. This represents a huge increase in entropy. Thus, the purpose of life from a thermodynamics view can be seen as essentially to increase entropy, but the formation of life in the first place is very much not entropically likely, and tremendous work had to have happened for it to have overcome entropy in the first place. If this is true, and it most certainly is, life, for it to have formed, the necessary concentration of such sparse basic building blocks would seem to be a complete impossibility. It would seem to be on the same level of likelihood as dropping a box full of computer parts on the ground and then opening it to find a fully assembled computer inside. In the case of life on early Earth, it was even more hopeless. The computer parts, or prebiotic organic compounds, were not even in a box together. Barring the orbital bombardments, they were most likely spontaneously created by sequences of low probability chemical reactions across wide stretches of volume in weight percentages ranging from 10 to the negative 9 in the ocean to 10 to the negative 3 in shallow lakes in the absolute best case scenario, in the case of formamide. A weight percentage of 3% to 33% is required to enable the polymerization of formamide into all the nucleobases necessary for DNA and RNA synthesis. For prebiotically relevant lipids to form protocellular vesicles, the critical accretion concentrations needed range from, in the case of decanoic acid, 100 millimolar at the high end, to phospholipids at 10 nanomolar at the low end. But all of the identified mechanisms for prebiotic synthesis achieve concentrations many orders of magnitude lower than that for each of the studied lipids. These fundamental building blocks that certainly did exist all have the same common theme of inadequate concentration. So why are we here? Without organic compounds in any significant quantity, how did life form? There have been experiments done throughout the years to try to explain this. The most famous one being the Miller-Urey experiment of 1952, where American chemist Stanley Miller and his PhD advisor Harold Urey passed electricity stimulating lightning through a mixture of ammonia, methane, water vapor, and hydrogen above a volume of liquid water simulating an ocean. The gas composition was what was thought at the time to be the atmosphere of early Earth. It was also assumed that lightning was incredibly common in the hellish environment of early Earth. The results were that a solution of organic compounds were formed in the water at significant concentrations. Although they were largely right about the lightning, the atmosphere at the time was actually mostly just carbon dioxide and nitrogen, but the experiment has been redone and the results are less promising but similar. The results at the time supported the Oparin-Haldane hypothesis, also known as the primordial soup theory of the origins of life, where life formed by simple molecules reacting over time, that was first proposed in the 1920s. Other problems with the experiment are that the resulting organic compounds are artificially concentrated within the flask, whereas in nature they would be spread widely across the environment and not accumulate, and they would be destroyed by UV radiation over time. The idea that the first life form was not a free-floating membrane-bound cell was popularized by American biochemist first Sidney Fox in the 1950s and later by Carl Woese in the 1970s, with their ideas of protenoid microspheres and the prebiotic progeny concept respectively. Carl Woese was most known for defining the third branch of the tree of life, archaea, as separate from prokaryotes in 1977, alongside George Fox. This concept would serve as a crucial foundation for future theories. A massive leap in progress was made in 1981 when Jack Corliss, an American oceanographer and geologist, first proposed that hydrothermal vents at the bottom of the ocean were the birthplace of life on Earth. Jack helped discover them in 1977, and ever since they have been the focus of most theories. The large black plumes of smoke emerging from the cone-shaped columns at the bottom of the seafloor were discovered at the Galapagos Rift at 2,500 meters depth with the submersible Alvin. They came to be known as black smokers. In 1988, Gunther Wasserschauser, a German patent lawyer with a PhD in chemistry, who was encouraged and supported by the Austrian philosopher Karl R. Popper, shook the field with his iron-sulfur world theory. He posited that life began as a series of metabolic reactions on an iron-sulfur mineral surface that catalyzed the reactions of early life. In 1993, Michael J. Russell, a British geochemist, took Wasserschauser's iron-sulfur world theory a step further and envisioned the first life as an iron sulfide bubble membrane formed on rock fractures at geothermal vents, where the confined space would enable organic compounds to accumulate from the vents and later be created by catalytic reactions of carbon dioxide, carbon monoxide, and formaldehyde with iron and sulfide bearing minerals. Although he did not use the term LUCA at the time, this would become the most modern theory on what LUCA would have looked like. Not a free-floating cell, but a bound iron sulfide membrane. The term LUCA, or last universal common ancestor, was later coined in 1997 by Patrick Forter, a French microbiologist. The current theory for LUCA is that eventually enough lipids would accumulate inside this iron sulfide scaffold-like environment that they would be able to form a free-floating lipid membrane as we know all cells to be surrounded by today. This origin of membranes would be consistent with how they currently exist today, as there is no genetic code for the membranes of any cells. They are kept from their parent cell as it splits into two, and lipid molecules are slowly added to the membrane over time as the cell grows until it is large enough to itself split. There was, as there always is, still something missing to this emerging theory. The concentration problem of life was still seemingly insurmountable. Organic compounds leaving the mantle into an iron sulfide bubble would still be in concentrations too dilute to have meaningful chemical reactions to polymerize and form more complexity. That missing piece would not be figured out until the start of the second millennia. Dieter Braun, a German biophysicist who at the time was a postdoctoral scholar at Rockefeller University, proposed the solution in 2002 with his paper Trapping of DNA by Thermophoretic Depletion and Convection, published with his postdoc advisor mathematician Albert Libschaber. In it, he argued that thermophoresis is the solution to the concentration problem of life. Where the other prior theories of evaporation of ponds, surface active minerals, or concentration from eutectic freeze-out failed to mesh with the hydrothermal vent theories of life's origin, thermophoresis, a lesser-known fluidical phenomenon, fits perfectly. Thermophoresis, also known as the Ludwig-Surret effect and thermodiffusion, is the fluid dynamics phenomenon where the components of a fluid separate from each other across a temperature gradient from hot to cold. Each chemical species within the fluid separates from other species and concentrates on either the hot or cold side depending on a number of conditions, including but not limited to mass, moment of inertia, size, heat of transfer, hydrogen bond network, cross interaction, ionic strength, and interfacial tension. Braun's idea was that dilute organic compounds would be concentrated by a combination of thermophoretic and convective flows in rock fissures at geothermal vents. The hot side would be the side of the fissure closest to geothermal activity, and the cold side would be that closest to the ocean. Over time, organic species would be trapped thermally, polymerized, and due to the concentrating power of these effects being more pronounced for longer chain organic compounds like DNA and RNA, this would naturally lead to a snowballing effect, leading to greater complexity and eventually life. Through the past 2 decades, Braun's work at Ludwig Maximilian University in Munich, Germany, has come closer and closer to fully experimentally proving how life began. The types of microfluidic experimental setups he has created to try to explain fundamental questions in the field have become widely influential. He has shown that thermophoretic and convective flows concentrate and polymerize nucleotides into DNA and RNA, and he has experimentally shown the formation of Lucas's speculated iron sulfide mineral membrane. What really drew me to his work in the first place was the work he did with his colleagues from his university on the emergence of pH gradients under these conditions. All organic life depends on the formation and depletion of pH gradients. pH is simply defined as a measure of the acidity or basicity of a solution. In biology, these gradients form across membranes, where chemical energy is used to pump protons, positively charged hydrogen atoms, across to form a lower pH acidic region. These protons then flow through proteins called ATP synthases to turn them like a motor to react adenosine diphosphate and phosphate to form adenosine triphosphate, ATP, the energy currency of life. This ATP energy is used by biological processes breaking off a phosphate to release chemical energy, and the ATP becomes an ADP and the cycle begins again. Most frequently, Dr. Braun has even experimented with the formation of a membraneless protocell. There are still many questions to be answered on how life formed, but within our lifetimes, I am confident we will have had an experiment that will simulate the most plausible conditions for the formation of early life, able to create a bound mineral-based synthetic life form akin to LUCA, and even be able to transition to a free-floating membrane-bound cell. Thank you for watching my video, and please like, subscribe, and turn notifications on so you don't miss my next video. I'll try to make it come out by the end of the year, and Dr. Pepper will appear in your fridge if you do as I ask, for real, no kizzy.